My name's Asha Bryan. I'm a scientist and director at ATD Bio.
My name's Tom Brown. I'm a scientist and a director of ATD Bio. ATD Bio was founded in 2005 by my parents, Professor Tom Brown and Dr. Dorcas Brown, both experienced nucleic acid scientists. I remember my parents bringing home HPLC traces of synthetic oligonucleotides on old-fashioned chart recorder paper over 30 years ago. I've always been surrounded by oligonucleotides through my parents' work. But in the present day, the potential of oligonucleotides to transform society in many different ways is incredibly exciting. Whether that's bringing forward the end of the pandemic, mRNA vaccine development, diagnostics for other diseases other than COVID, curing genetic and other diseases, whether that's through therapeutic oligonucleotides or gene and genome editing or something else, and also the development of new materials. We all have DNA in almost every cell of our body, and human genomic DNA is around 3 billion base pairs in length. Oligonucleotides are small pieces of synthetic DNA or RNA, ranging in length from typically around 3 to 200 bases. The oligonucleotides that we make routinely are often in the range of around 15 to, say, 120 bases. So despite that large discrepancy in size between synthetic and genomic DNA, the ability of synthetic oligonucleotides to hybridize or bind to genomic DNA is the basis for many of their useful properties. And oligonucleotides have uses in lots of different fields, from diagnostics to therapeutics and other areas as well. It's quite strange for us as oligonucleotide manufacturers to see oligonucleotides being mentioned literally on the front pages of national newspapers, as has been the case here in the UK during the course of the COVID-19 pandemic. And the reason for that, of course, is the role of oligonucleotides in COVID-19 testing. So the most accurate and the most sensitive methods of COVID testing require synthetic oligonucleotide primers and probes. So that's either primers and probes for PCR or for other isothermal amplification methods.
We produce oligonucleotides which are between about 3 and 200 bases in length, but most commonly between about 15 and 120. Our traditional speciality is in the synthesis of more complex oligonucleotides which incorporate chemical modifications which are not found in natural DNA. Some really typical examples of the most common modifications that we might incorporate are fluorophores and quenchers, which are very common in diagnostic oligonucleotides. For therapeutic and in vivo applications, modifications to the phosphate backbone, the sugar moiety, or the 3 and 5' termini of the molecule to increase stability to nuclease enzymes, or to help with delivery to specific organs within the body or into cells. Oligonucleotides are synthesized using a highly optimized, automated solid phase approach. We synthesize them most often in the 3' to 5' direction, which is actually opposite to the direction that biological polymerase enzymes work in. After the oligonucleotide synthesis, the first step is deprotection of the protecting groups on the phosphate backbone and the nuclear bases. After that, we usually perform some basic cleanup steps before injecting the oligonucleotide onto the HPLC system. But I think the most important step that we perform before purification is rigorous analysis of the crude product. And the reason for that is that the quality of the oligonucleotide synthesis is probably the single most important factor in determining the final purity of the product. In many ways, DNA and RNA are very stable molecules, especially in their native environments, and that's partly because they have the advantage of about 3 billion years of evolution. Having said that, especially when they're in solution in an uncontrolled pH environment, there are some cleavage and degradation reactions that you have to be aware of. In biological foods, they can be degraded very rapidly by nuclease enzymes. And of course, when you're dealing with chemically modified DNA, there's a whole host of possible decomposition side reactions which you have to take into account depending on the nature of the modification. So the first step after purification is to separate the oligonucleotide from the buffer which it's eluted in. So the volatile solvent components of the buffer can just be moved by evaporation. You then usually require a desalting step to remove the ion pair components of the buffer, unless you happen to be using very volatile ion pair components. So the 3 main methods of desalting are size exclusion chromatography, where you're relying on the smaller salt components being trapped in a size exclusion resin and the larger oligonucleotides passing through. Also dialysis type methods, which relies on the greater ability of small salt-like molecules to diffuse across the membrane, and also precipitation methods, which rely on the differing solubilities between the ion pair components of your buffer and the oligonucleotide molecules. So there are two HPLC chromatography methods which are used very commonly for oligonucleotide purification. The first involves a reverse phase stationary phase, so silica functionalized with alcohol chains, and that works by a combination of polarity and hydrophobic effects combined with ion pairing because there is an ion pair component in the buffer. The second really common method is just using pure ion pairing reactions. In that case, you would have a stationary phase which is functionalized with cationic groups, so something like quaternary ammonium, and that method relies purely on electrostatic ion pairing interactions between the cationic stationary phase and the anionic oligonucleotides. To highlight the magnitude of the increase in scale that we've tried to achieve, typically before the pandemic, the most common synthesis scales that we would work on were the 0.2 micromole and the 1 micromole scale. Although we've always done some larger scale synthesis, those were by far the most common scales that we would work on. And that would typically yield between about 0.5 milligram and 5 milligrams of material after purification. Recently, we've been supplying certain sequences on the scale of tens of grams, so that's about a 10,000-fold increase in scale. So obviously achieving that has been a really major technical and logistical challenge. We've had to repurpose our research and development site in Oxford as a production line, and we've needed new equipment, new staff, and completely new procedures. I think the solid phase oligonucleotide synthesis process is already a highly optimised and quite an impressive feat. One major challenge as demand for oligonucleotides continues to grow, especially really large-scale oligonucleotides, will be how can we make this process even more efficient, especially with regard to reagent consumption. I think one example which is really commonly cited is that if therapeutic oligonucleotides were to take off in a really major way, the current worldwide supply of acetonitrile, which is one of the major solvents used during oligonucleotide synthesis, would be completely exhausted before we could reach the capacity that we need.
ATD Bio's future is incredibly exciting. So we'll continue in our core business of making highly complex, high purity oligos for our customers all over the world who depend on us for that. As a result of our large expansion in synthesis capacity, we'll continue to make oligos for diagnostics on a large scale. We'll also be able to make oligos for therapeutics, so both for initial development work, but also potentially moving into early stages of clinical trials. We expect there to be a continued need for COVID-19 testing in the short and medium term, whether that's for surveillance, monitoring levels of the COVID-19 virus in the population, or whether it's monitoring the efficacy of the vaccines, the various vaccines, or also monitoring the evolution of the virus, the different variants of the virus, and also for future preparedness for future viruses that may come along. We also expect there to be approval of new oligonucleotide-based therapeutics and possibly the first real blockbuster oligonucleotide drug, and a continuation in interest and surge in interest in therapeutic oligonucleotides across all sectors, including from the pharmaceutical industry. We expect there to be further development in gene and genome editing. We're really excited about new sequencing technologies which will provide further information or new information, for example, on the genetic makeup of tumours. We're sure there will also be new uses of synthetic oligonucleotides we haven't even imagined yet.